And I think we left off with phosphorus being the trickiest in terms of for the plant to get access to phosphorus. Most plant physiologists would agree that this mineral is challenging. It's challenging to supply enough in agricultural fields or in, when you're growing plants for profit. And to keep your soil at the right pH is important. And so managing all of that so that they can get access to phosphorus is a big deal. And we looked at this last time where some plants, not all, but some plants have evolved the mechanism to more deliberately get access to phosphorus, where you have citrate being released, for example. So that's something that they make and then they excrete it out. And as the citrate is interacting with some of the bound phosphorus, it helps release the phosphorus into the water solution of the soil, and then the plant can take it up. But you see that it's usually dependent on, spending some ATP in order to get a proton gradient so that those protons can drag in the phosphorus through that symport process that's being shown here. And then some other plants might also have this option of making acid phosphatase enzyme, excreting that out. And again, it helps solubilize the phosphate off the soil particle. But again, you see there's a cost. They have to expend ATP to get that proton gradient to bring in that phosphorus. And then the CO2 idea, that's more like, plants aren't really, they just do that all the time because they're respiring. So just like you have to breathe out CO2, plants are doing that too. And so there's that action. If there's enough CO2 in the vicinity where there's some bound phosphorus. So there are some options available, but it's still a problem. And I wanted to show you this idea. This is actually my former postdoc advisor, Dr. Linda Kinkle. So she's been doing a lot of work looking at microbes in the soil and ways that we can use microbes in the soil to help benefit plants, whether to protect them against disease or help get them access to minerals and other nutrients. And so I put this in here because this particular blurb is talking about phosphate availability. And so she actually, she's been an amazing scientist for decades. And she was very frustrated with the idea that, she's doing all this beautiful research, getting great results, and just, writing up, publishing, getting grant money. And yet it never seemed to have translated into the real world. Like she's got all these great ideas and great results from all these experiments, but then no one is like saying, here, if you buy this and use this, it's going to work. It just seemed to stop. And so she actually decided to start her own business and get what she's learning and what she's finding out into the real world so that people can actually benefit from it. And so I thought she just did that just within the last couple of years. So she's at the end of her academic career, and she's apparently not run out of steam, and she's turned her attention to, now I want to take what I've been learning all these decades and get it out there more widely. And so one of the things they're focusing on in this is ways to augment soil with key microbes in order to increase phosphate availability. And that's their main focus, is using beneficial microbes. And with some of the experiments that they have done before they formulated their final product, they were showing, great increases in phosphate solubilization. I have a hard time saying that. So 158% increase, that's dramatic. And so that definitely will catch the eyes of growers who are trying to grow plants in, acres upon acres and trying to get beneficial, yield from them at the end of the growing season. So this is one of the things that they work on, but it's not just phosphorus that they're zoomed in on. They are looking at other ideas too, but I thought that was an important thing to see. And then just in general, this kind of tells the story of how she decided to go into business and start a business with the idea that she wants to take what she's been learning through all this great research and start getting it out there for real world applications. And I think that's just awesome to see that connection and trying to connect out into getting this to happen in the real world. This was a paper that was published back in 2020. And I wanted to bring it up as we're talking about phosphorus because now they're starting to look more carefully at mycorrhizal fungi and like, what are all the roles that mycorrhizal fungi play in the soil? You know, initially it was, well, they help the plant get access to water. They help the plant get access to minerals. Maybe they defend the plant a little bit through biocontrol mechanisms. Here, they're actually looking at the idea that the mycorrhizal fungi seem to be controlling who gets phosphorus and how much. So they are actually, it's hypothesized that the mycorrhizal fungi have a lot more control on what's happening in terms of a forest ecosystem, in terms of how the nutrients are flowing. And so when you look at a forested ecosystem with a mixed hardwood forest or some conifers mixed in there too, the idea that these mycorrhizal fungi are connecting these plants underground through their mycorrhizal fungal hyphae connections. And so this is something that is being heavily studied probably within the last decade. It's really gained a lot of interest. And if mycorrhizal fungi are connecting all these plants underground, and if they're being used as a way to transmit water and nutrients, is it just a passive thing or is the fungus more involved than what they initially thought? And the answer seems to be so far that the fungus is more involved and has a greater say in how those nutrients and chemicals are flowing from tree to tree to tree, for example, in a forested ecosystem. In this particular study, they were looking specifically at phosphorus. And a really nicely done study. And they were showing how kind of like in economic trade policy, where the price goes up when demand, when supply goes down, right? Then you raise your price and so on and so forth. And so they talk about trade theory in here and economic, what makes sense economically. And then they kind of apply those concepts to the mycorrhizal fungi's ability to control when the plants are getting phosphorus and how much are they getting. And so really, really interesting research going on. And I think there's going to be more of this type of research where we're starting to realize the mycorrhizal fungi are major players. They're not just kind of passively letting things flow through their hyphae. So lots of fun. And then this one is pretty amazing. This study was looking at mycorrhizal fungi and then bacteria that skate along the surface of the hyphae. And that together they're helping break down phosphorus and make that available for plants. And so this was an amazing study. And so mycorrhizal fungi, again, they dropped the V off here. I don't like that. Keep the V, it's easier to say. So the VAM fungi were gaining access to nutrient patches. And here they looking at phosphate solubilizing bacteria and the fungi in bespoke microcosms. So this was a controlled study, and they were looking at what's going on. And it said the hyphae can transport the bacteria to the patches of phosphorus, and then their actions allow them to break it down. Bacteria move in a thick water film formed around the fungal hyphae. However, the bacteria cannot be transferred to the organic phosphorus patch without an energy source in the form of hyphal exudates. So what they're saying there is that it looked like the fungi were having these bacteria skate along their surface. As the bacteria are moving, the fungi are feeding them a little bit of carbs probably to keep them going so the bacteria could stay active, get them to the phosphorus patch, and then they both like break it down. And then of course some of that gets sent back to the plant through the mycorrhizal fungi. And so they even took, let me see if I can get the video to show. No, what am I doing? I want this. Yeah, I'm gonna do the big, get this down. So there's no narration, it's just showing the video. So Pseudomonas putida is the bacteria in liquid films around the fungus, overgrowing the surface of potato dextrose agar. Real-time video. So they're zinging along. Looks like the what's that highway system in Germany where it goes really fast? What's the name of that highway? Where like you can go as fast as you want. They're zipping along. That is fast. That's real-time movement. That's not sped up. So yeah, they're just zinging along. So there's like a thin water film along the hyphae, and they're just skating along like crazy. Isn't that cool? And so as they're moving along, apparently the fungal hyphae are feeding them, keeping them going so that they can make it to the nutrient patch and start helping to break down the phosphorus. So all kinds of cool research going on with mycorrhizal fungi and their associated microbes. And now I want out of there. Okay. Let me back into sharing. Okay, so lots of effort is going into trying to understand phosphate solubilization, ways for plants to get easier access to it. There's also a concern that we're running low on natural phosphorus, like they, mine phosphorus in areas that they've been mining for phosphorus. The phosphorus amount is decreasing. There's a lot of concern about that, so that's another issue to consider, why they want to make sure that all parts of phosphorus in the soil system are getting some use, hopefully, and won't go wasted. Potassium. So NPK, when you go to buy a fertilizer at a store, is always the NPK, and then the K is the potassium. In agriculture, in big fields, they usually apply potash, which is K2CO3. That's commercially sold. You need a lot of potassium for a plant to grow very luxuriantly through its whole life. Luckily, it's very soluble in the soil water. The problem is if you have sandy soil, the potassium tends to leach down with the water through that really sandy soil. The particle sizes are so big that it doesn't retain water very well. And so in sandy soils like we have around here, we have a lot of sandy soils in this area, which is why there's a fair bit of potato growing around here, because white potatoes do enjoy sandy soils. They like that. But anyways, very soluble in the sense that it's easy to go through cation exchange, get the potassium into the soil water, then the roots take it up. It's also highly mobile in the plant. So once it gets into the plant, it can get redistributed to younger, more vigorous growth if necessary. Look for mottled chlorosis or necrosis. Also look for yellowing or browning of the tips and margins of the leaves. That's often a good giveaway. So when I see margin and tips going brown or black, I know that's probably potassium. In the field, if your plants like wheat and barley and so on, if they get deficient in potassium long enough, it causes weakening of the stem. And then you'll go out to your field and see that they've totally bent over. And now all the area of the plant that has the grains and developing grains and seeds, it's on the ground now. And that is terrible if you are a grower because your machines don't harvest when they're bent over like that. It's incredibly challenging. So it's very difficult. And of course, if you have your grains on the ground, they're going to get a much higher chance they're going to get attacked by fungi and bacteria and eaten up before you can harvest them. So this is this lodging, they call it lodging of plants. That's super serious. You do not want that to happen. A couple of other minerals I'll mention. Sulfur, that one's usually not an issue. It's unusual because some of our amino acids require sulfur. So we definitely need it. All organisms need it. So cysteine and methionine are two amino acids that have sulfur as part of their chemical structure. Generally, it's not a problem in terms of deficiencies because so much of our pollution has enough sulfur in it that that's fine. One volcanic eruption can provide a lot of sulfur to a large geographic regions. So again, it's not a huge problem. It's part of thiamine, biotin, coenzyme A. So all of those are necessary for reactions and and then the iso and thiocyanates are secondary metabolites that plants make. So if you think about mustard or horseradish, that kind of pungent taste that you get when you bite into broccoli or cabbages or turnips, that is due to high levels of isocyanates or thiocyanates or isothiocyanates. You tend to see general chlorosis, and because cysteine and methionine are very important amino acids that get put into a lot of proteins. If you have a deficiency in sulfur, you tend to see protein synthesis crashing. Once it gets taken up by the plant, it is immobile. So younger growth will suffer if there's a deficiency, and you'll see symptoms in the young growth first. That's the area to look for. By the way, so on these slides, I do talk about symptoms, but I won't ask you, like, tell me what deficiency symptoms are for sulfur. Like, I won't do that because so much of the time, some of the symptoms for one mineral are the exact same for another mineral or close to it. So there's no easy way to be precise about that. But I will be asking about, what does general chlorosis mean? What does mottled chlorosis mean? What's intervenal? So knowing the idea of it, but you don't have to memorize deficiency symptoms of the different minerals. Calcium, usually not a problem. Very important in the process of cell division. So when cells are going through reproduction and dividing, you need calcium as part of that makeup. It is also helping with neighboring cells to be able to adhere to each other. So the adhesion processes of tissue. And then this other idea, second messenger. We're going to learn down the road a little bit here that calcium is very, very important in providing information to cells. And so as the plant is assessing its environment, it needs to be able to assess the environment, but then also shift its gene expression to respond to its environment. And in order to do that, so assessing is one thing, but then you have to be able to use that information to change your gene expression. And calcium is part of that process, very heavily part of that process. And so we'll talk more about calcium in that role as a second messenger, helping the cells shift their behavior and gene expression. Now, this one tends to have pretty dramatic symptoms. You see that the apical meristem region of the shoot in particular really slows way down. So you get super stunted growth. And then any new leaves that form under a deficiency, they look pathetic. They're curled up, they're wrinkly, they never quite fully extend into their full form. And they usually start out yellow and then they quickly go brown or black. If you have a calcium deficiency in hydroponic systems, you can see the roots easier in hydroponic systems. And again, you see really poor root growth. I almost see when I do calcium deficiency experiments, it almost, at least to my experience, is calcium deficiency hits the plant harder than nitrogen deficiency, even though plants need nitrogen in higher levels than calcium. If you put a plant under calcium deficiency, oh my gosh, they look really bad really quickly. And they never seem to recover from it. It's immobile once it gets into the plant. So young leaves, young tissue really get hit hard. Magnesium, very, very important. Required in large amounts by the plant. Mg2 plus is usually the form. It can be common to be deficient if you don't have a good control on your soil pH. So more acidic than 5.5, or if you have really sandy soil, it's going to leach through with the water. It's super important because chlorophyll can't work without it. It needs magnesium. Ribosome function, it helps to be an activator. It activates enzymes, including two huge ones, rubisco, which is the most abundant protein on the face of the planet, and then phosphoenolpyruvate, which we'll talk more about. It helps with ATP. It has a pretty classic symptom where you'll see interveinal chlorosis. And so often, if I see a plant going through interveinal, my very first thought is magnesium. There are other symptoms that do interveinal, but because plants need magnesium at such high levels, I tend to suspect magnesium is the culprit or lack of magnesium. Once the plant takes it up, it is mobile. So young tissue will get fed, older tissue will start to suffer because they gave up the magnesium. So chlorophyll is the pigment necessary for photosynthesis. Here's a mock-up of it. And you can see how magnesium is chelated right in the so-called head region. This is the porphyrin head region of chlorophyll. And you can see magnesium is chelated right in the center. So this is the porphyrin head region, and then this is the phytol tail region. We'll learn more about chlorophyll in just a little while. And so all chlorophyll needs magnesium. If any of you have taken animal or human phys, that might look a little familiar to you, that structure, because hemoglobin is very similar in structure to chlorophyll. So instead of magnesium, you have an iron, but look at the porphyrin head region, pretty similar. So it's kind of neat. Whoops, kind of neat that you have that. I don't know why that's doing that. It was doing that the other day too. It won't let go of the writing. So interesting comparison there. And I didn't know this until I was in grad school. Magnesium is actually necessary for fully functioning ATP. I don't remember learning that. And maybe they didn't know that back when I was an undergrad, but I don't know if they teach that now. Has anyone taken phys classes, human or animal phys, where they might have talked a little bit more? Yeah, I mean, I'm like, what? So magnesium is part of functioning ATP. Under physiological conditions, so in the body, ATP always forms a complex with magnesium. Most enzyme binding sites for ATP actually bind magnesium ATP. This is one reason why magnesium is an essential nutrient for all living cells. So once I learned that, it's like, ooh, now I understand why, people are, because I was reading a lot of papers at the time in grad school where magnesium deficiency in agricultural soils was becoming a concern. But they never really said why. And then I learned this. So because of this, I choose to take a magnesium supplement to help, because I know my diet isn't the greatest often. So, but you got to be careful with what type of magnesium you take, because the cheapest bottle of supplements, magnesium oxide, that just goes right through you. That's more for helping you stay regular and going to the bathroom on a daily basis. So that won't really help you. That just goes through you. You have to get the chelated versions of magnesium for it to get into your cells and be bioavailable. Why is that doing that? It won't even let me pick the eraser. That's annoying. All right, sorry about that. I can't erase it. It won't let me. So micronutrients, I'll just say a couple things. There is a table in the textbook if you're curious about micronutrients and their impacts. But there's only a few I want you to think more deeply about. So, well, iron is probably the biggest one. Iron is common in the soil in two forms, Fe2 plus and Fe3 plus. Fe3 plus I put in parentheses because that form is virtually unavailable to plants because it's insoluble. In fact, if you've ever had to try to make a solution in a lab that has the ferric, so Fe3 plus is the ferric form of iron, takes forever to dissolve. You have to heat that solution up and stir and stir and stir. And usually I can't even get it to all go into solution unless you chelate it somehow. So anyways, Fe2 plus, that's the form that's more soluble. So that's typically what the plant will have better access to. However, the plant needs it. Of all the micronutrients, it's required in the largest amount. In fact, there's some physiologists that say maybe we should put it with the macronutrients because it's such a demand by the plant. They haven't done that yet. They kind of are slow to move something. So that's such a paradigm. But anyways, it's very valuable for the plant because it helps the plant do redox reactions, which the plant is doing every minute of the day. And then in order to make chlorophyll, iron is involved in the biosynthetic pathway for that. And chlorophyll, of course, is needed in huge amounts. This one also will show intraveinal chlorosis. So between magnesium and iron, those are usually your two thoughts. I tend to go with magnesium when I see intraveinal, but I know that it could be iron as well. It is immobile once the plant takes it up. And so younger tissue suffers first if there's a deficiency. And if you go a little too acidic in your soil system, it actually can the plant gets too much iron. Too much of anything can be bad for you. And so this idea that they get too much iron and it starts to become a toxic issue. That's true for humans too, more so for men than women, that they can get iron toxicity. So it's needed in large amounts. Deficiencies in different soil systems is fairly common, at least for stretches of the growing season. What tends to happen is you can get, especially the ferric form, that's not easy for the plant to get. It's very happy to react with other chemicals and then precipitates. And so if you have that happening, particularly if your pH is closer to 7 or you get up into above 7, you tend to see the ferric form getting really insoluble. It won't be available. So then what they've found is that some plants have evolved mechanisms to grab iron, especially even like the ferric form, and they do that through chelating it. And so what they do, like in the lab, what we do to chelate iron and get it more soluble is we add EDTA to the solution, or you could add in caffeic acid. And plants actually do that too. They will secrete out caffeic acid with the goal of getting access, especially to the ferric form of iron, and it chelates it and stabilizes it, and then they can bring it into the roots. So here, this is just trying to show the ferric form, Fe3 plus in the lower image here is chelated. So this is DTPA chelating agent. So you can use DTPA in the lab. And if you have to work with ferric chloride, for example, or something like that. But plants can do it. So plants are secreting caffeic acid for iron. And then we learned earlier, where'd that one go? You know, for phosphorus, they're doing citrate. And so again, some plants have this ability to go ahead and secrete out chemicals that will give them that access to iron in this case. So with the caffeic acid. So caffeic acid, the structure is being shown there. Another thing that they can do, they have these special cells that are called transfer cells that are lining on the surface of their roots. So you can see discrete areas, patches of the epidermal tissue will have these so-called transfer cells. And it seems like that's the site of where iron is coming in easier for the plant. But that is still under a lot of study. They don't know exactly how it works. But it seems like some plants do that. Some plants will enhance or ramp up protons in their immediate environment by their roots. So if they're going to do that, that means they're expending ATP, but they'll pump out extra protons, and that seems to also help solubilize the iron. The caffeic acid idea that I told you about. Once they get the iron to the root, they usually will act on it, and then they'll be able to bring it in and have that into their body. And so usually the chelated form of iron gets released from the chelating agent, whatever that is, and then they can take it up. So there are some strategies that have evolved through this process of, you know, having that iron in a less available form. And so there are strategies that have evolved. Another strategy that's been heavily studied in the grass family, Gramineae, is the special compounds that the plant makes called phytosiderophores. So phytosiderophores are a special class of chemicals that will act kind of like chelating agents again. They will bind especially the ferric form, Fe3 plus. And it's kind of unusual because these chemicals will latch on to the iron and then everything gets brought in, including the chelating agent. And everything just gets reabsorbed by the root, and then the root will release the iron and have it sent on its way through the plant. But so the grasses were the first to discover the phytosiderophores. There, so far it's kind of limited. They don't see that showing up in a lot of other groups of plants. By the way, if you drop off the word phyto or that, anytime you see PHYTO in science, that's plant or plant-like. That's what that means. Siderophores, if you just drop off the phyto, you just say siderophores, that's also a thing. And so bacteria make siderophores. We know that they make siderophores, and again, to latch onto iron. So what I learned not that long ago, from one of our physiology faculty, is that if you are sick with a bacteria pathogen and it's making you sick and you pop a fever, and I think most medical doctors know this now, they actually say, let your fever go. Like, don't immediately treat it. Don't take ibuprofen or aspirin or whatever to bring it down. And unless it goes, they say, once you go over like 101.5 and onward, yeah, maybe. But otherwise, they say, let a low fever run. And the reason for that is bacteria in your body that are causing problems, they usually pathogens produce siderophores because they're in your body and they're hunting for iron of your body. And so they're attacking your body for iron, and they release these siderophores in order to do that. And so if you pop a fever, it causes your body to make special proteins that latch onto iron and keep it away from the pathogen. And so popping a fever is actually beneficial to a point. I mean, eventually if it gets too bad, it's pretty miserable and it can get dangerous. But um, when my boys were toddlers and I took them to the doctors with fever, they were like, yeah, just let the fever run, monitor it, but let it run. Because they were aware of this idea that your body is actually making proteins to help keep the iron away from the pathogen. I think that's why my grandfather was 100% Finnish. Anytime he started to feel punky, he'd go and take a sauna, really hot sauna, to elevate his body temp, like, artificial fever. And so even though he didn't know any of this, I wonder, he swore that it helped him feel better quicker. So maybe it was doing that. It was keeping the iron away from the pathogen. So this is just a mock-up showing the idea of this. Here in the middle here, you have this Fe3 plus chelated. They're trying to show that it's chelated, and then it's getting acted on and getting into the Fe2 plus form, and then the Fe2 plus form is getting taken in. So they've, looked to that level of detail on how is the plant doing this. And then they have the siderophore idea, and then they're showing that the whole thing comes in. So I don't think this is accurate. Do you see the name of that transport protein? I don't think a transport protein is really called yellow stripe 1. I think that was the illustrator, like that was a placeholder for what the name was supposed to be, and they never replaced it. Because this is the yellow stripe. My guess is the illustrator didn't finish the illustration. I've never heard of an enzyme called yellow stripe 1. Okay. Then, besides macronutrients, micronutrients, there's so-called beneficial elements. Now, this is a bit nebulous, and people argue about this. So I'm going to give you a list, and you don't have to memorize this or anything, but just be aware that these are often listed, but people argue about these. And so I'm not going to really belabor the point. Beneficial just means that they don't make the list of macro and micronutrients because not all plants need them. It really is driven much more by the habitat the plant lives in and how they've adapted to that habitat. So some habitats are silicone rich, and plants have taken advantage of that, and you'll find silicone in their cell wall, for example. So again, take this with a little bit of a grain of salt, because this list keeps changing over the years. But there is a thing that people call beneficial elements. You'll hear that term. This I found from a different resource, and it just, if you're curious on what can cause deficiencies in these different minerals, they give a little bit here of what can happen. This isn't exhaustive, but it's just trying to show, if you have calcium deficiency, it's probably too acidic or too alkaline, for example, soils. So it's just kind of a handy table. And then the last few slides are more just for thinking about if you have too much of something. So if growers misjudge how much nitrogen to add, which historically that's not all that uncommon, they put too much nitrogen into the system. You tend to see lots of growth in the shoots, but not much in the roots. So if your biomass like potatoes, you want the potatoes to grow, not the leaves to grow. I mean, you need the leaves to grow, but you definitely want some good root systems too. Same with sweet potatoes. You want good root development. So too much nitrogen and it tends to overfavor the shoots. What tends to happen is the leaves get really luxuriant, but the stems not so much. And so because you have all that extra leaf biomass and the stem isn't quite as strong as it should be as normal, it tends to allow the plant to flop over or lodge. If you see curling of the leaves, that usually means that there's too much ammonium, if that's how the source that you fed it. And it slows down flowering in agricultural crops. So not ideal, especially if you have a temperate growing area like up here, where winter is, barreling down on you as a grower. You don't want onset of flowering to be delayed. So too much nitrogen is a thing. It's not all that uncommon. Same with, sometimes they overdo it with phosphorus because they worry about the phosphorus being an issue. And that can get your roots going, but then your shoot, your stem and leaves they're not growing as fast, but your roots are. So again, you get this imbalance in the different parts of the plant. Phosphorus can be a problem if you get too much put into the system. It actually interacts with the plant's ability to take up iron and slows that down, as well as manganese and zinc. And so soil scientists and plant physiologists have worked pretty carefully at trying to figure all this out. Like if you have an excess in this nutrient, what does it do to some of the others? Does it make it harder for some of the other nutrients for the plant to get access to those? And the answer is yes. And it's really complicated, but they have a lot of this information out there now that if you overdo this nutrient or that nutrient when you're growing your plants, it's going to be kind of a domino impact where you'll see other minerals also being an issue for the plant now. And then potassium, if you put too much of that in, so your NPK, right? If you over fertilize, even in your plants at home, in your pots, you can get too much potassium and not enough magnesium, which again, pretty important idea here. You don't want deficiencies in magnesium and calcium, not a good idea. Okay. Minerals, very much needed. So that is the content on mineral nutrition and mycorrhizal fungi and beneficial bacteria. I did want to stay in the soil though for the next topic because we're going to talk a little bit more about nitrogen. And we need to think about some very special cases of how plants get access to nitrogen. So this is the idea of biological nitrogen fixation. So instead of the grower putting a bunch of nitrogen inorganic fertilizer onto the field, this is the idea that the plant is partnering up with a bacteria that can fix nitrogen. And this partnership is a very intimate partnership to the point where the plant lets the bacteria live in its tissues, in its root tissues. And the bacteria will get a nice little house in the root tissue and fix nitrogen. And then a lot of that nitrogen goes to the host plant. The bacteria also benefits, gets its own nitrogen too, of course, but it will also send a lot of that nitrogen to the plant. And this partnership has been heavily, heavily studied because like the holy grail is, can we create this partnership in other agricultural crops? So there's some key agricultural crops that do this naturally, and they are now on the path to make other non partnering plants to have this ability. So it's a hot topic right now in research. Synthetic biology is becoming heavily involved in this process because it's a lot of intricate pathways that have to just line up between the host and the bacteria. And so they're working really hard at this. So next week we'll get into looking at how does this partnership develop and then what exactly is going on to allow for this process to occur where the plant gets all this lovely nitrogen produced by the bacteria. But we'll start that on Monday since it's a big topic. So we'll quit a few minutes early here. Okay. All done, except I do want you to hand something in. So